Welcome to Prospective Doctors MCAT Podcast, brought to you by Med School Coach. Each week, Sam breaks down the highest yield MCAT topics so you can score as high as possible on test day.
Hello and welcome to Prospective Doctors MCAT Basics with your host Sam Smith. The goal of this podcast is to cover the highest yield topics on the MCAT and provide you with some sort of insight into the questions that the MCAT really likes to ask. This podcast is going to cover the adaptive immune system. This is the second part to a kind of a two-part series on the immune system. The last podcast I did was on the innate immune system. This, as I said, is on the adaptive immune system. The first thing I'm going to talk about is some general vocabulary terms that I'm going to use quite a bit during the podcast. And then I'll talk about the cells that make up the adaptive immune system. And then I'll break down the adaptive immune system into the humoral branch and the cell-mediated branch. And then at the end, I'll talk a little bit about memory B cells and memory T cells. This material is most likely to show up on one out of the four sections of the MCAT. That is the biochem section. Thanks as always for listening to the podcast. I hope it helps. All right, so last podcast I talked about the innate immune system. And I just want to remind you about the distinction between the adaptive and the innate immune system. So the innate immune system tends to be this super fast, non-specific response, whereas the adaptive immune system tends to be a much slower, more sophisticated, more targeted response to some kind of pathogen or some kind of antigen that has been recognized within the body, that the body recognizes as foreign and wants to get rid of. And so before I dive into the adaptive immune system, I want to go over a few vocab terms that you'll hear me use throughout the podcast that you really need to be familiar with. The very first one is antigen, and an antigen is just some kind of toxin or foreign substance that induces an immune response in the body. And typically this is a part of some pathogen. So the example I gave from last podcast was that some of the envelope proteins of the HIV virus can act as an antigen, whereas HIV itself is defined as a pathogen. So there's a little bit of difference between those two terms. The next term that I want to define for you is major histocompatibility complex. So there are two of these. There's a class 1 major histocompatibility complex and a class 2 major histocompatibility complex, and that's abbreviated MHC. So let me first start by defining Mhc Class 1 molecules. So Mhc Class 1 molecules are found on the exterior of all nucleated cells in the body, and essentially their job is to display antigens or proteins or parts of pathogens that have entered the cell. So what they do is they essentially take these viruses or whatever has entered the cell, and they are able to move that to the exterior of the cell and say, okay, hey, look what's inside of me, and kind of point that, towards other immune cells that can then recognize it. So take this example. Say I eat a jelly bean and I swallow the jelly bean, it goes into my stomach, and all of a sudden my friend says, hey, what type of jelly bean was that? And I'm like, shit, I forget, I don't know. So what I do is I make an incision and I pull that jelly bean out, and I stitch up that incision, and then I just set that jelly bean on my stomach where that incision was. And now my friend can come up and look at it and kind of inspect and say, oh, that was a blueberry jelly bean. And so in the same way, cells are able to take these pathogens that are inside of them, break them down a little bit, and then they can display the proteins on their surface. So the two big takeaways for Mhc Class 1 molecules is that number 1, they are on all nucleated cells, and number 2, they display antigens that are present within the cell. Major histocompatibility complex 2 molecules are a little bit different. So these are glycoproteins found on the exterior only of these professional antigen-presenting cells. And when I talk about the cells that make up the adaptive immune system, I will let you know exactly what cells are in the category of professional antigen-presenting cells. And so what they do is they show off proteins that are found extracellularly. So let's go back to my example of the jelly bean. So say I am in a lazy river and I'm floating around in a tube when all of a sudden I see a bunch of jelly beans just floating by me. And of course, I'm with my friend again, and I just grab one and I pick it up, and my friend goes, "What flavor is that?" And so, I just hold it up to him so he can see it, and he's like, "Oh, that's black licorice." And at that point, I obviously aim for the nearest trash can to throw the jelly bean away because that's the worst flavor of all time. And so this is just to say that Mhc Class 2 molecules are able to recognize pathogens that are outside of the cell. And then once they bind onto these extracellular antigens, they're able to show other immune cells these antigens and then elicit some immune response. So overall, these Mhc molecules essentially act to bind onto pathogens and show them to other immune cells that can then elicit a response. And this will become a lot more clear when I begin to talk about antigen presentation and talk about how that works. For now, I just wanted to introduce these terms because you're going to hear them a lot throughout this podcast, and it's obviously very important you know the difference between Class 1 and Class 2 of these Mhc molecules. You might see some kind of question asking you about that on the MCAT. All right, so let's get now into the cells of the adaptive immune system. So I have broken these cells down into 3 categories. That is B cells, T cells, and professional antigen-presenting cells. And in each of these categories, you have numerous cell types, especially in the B and T cells. You have these cells that are like T follicular helper cells, T follicular memory cells, and it breaks down into a bazillion categories. So what I really want to do is just hit on a few of the very specific, most important cell types that you'll see in these categories on the MCAT. So first off, B cells or B lymphocytes are produced in the bone marrow, hence the B for B cell, and their main job is to produce antibodies. And they also serve a secondary role, which is presenting antigens. So they are an antigen-presenting cell, but they're most known for their ability to produce antibodies. And we'll get more into this when I start talking about humoral immunity. In terms of T cells or T lymphocytes, there are 2 main types that I am going to talk about, and they have pretty different roles. So the first type of T cell is called a CD4 positive T cell or helper T cell, and they recognize peptides that are presented on Mhc Class 2 molecules, which, if you remember back to my definition, those are the molecules that are able to recognize extracellular antigens. And CD4 or helper T cells play a big role in orchestrating and organizing the adaptive immune responses. And they do this through helping B cells make antibodies. They induce macrophages to come and help with these different pathogens that are in the body. And then they also recruit neutrophils, eosinophils, and more cells. So you can kind of think of the CD4 or helper T cell as what its name says, as a helper, right? It's got all these different functions that kind of tie together different bits of the immune system. On the other hand, you have what are known as CD8 positive T cells or cytotoxic T cells. And this is the second kind of T cell that I want to talk about. And these CD8 T cells bind to Mhc Class 1 molecules. And remember, these are the glycoproteins that are able to bind to intracellular antigens. And essentially their role is to bring these antigens to the surface of the cell and kind of let the cell say, hey, I'm infected with this bad pathogen. Come help me take care of this pathogen. Come kill me, whatever this T cell has to do to get rid of this pathogen. So the role then of cytotoxic T cells or CD8 T cells is to recognize cells that are infected with some pathogen and actually kill those cells. In addition to killing cells that are infected with some sort of pathogen, whether that be a virus or bacteria or fungi, these CD8 T cells can also recognize cancer cells and kill them. So those are the two types of T cells that you really should know for the MCAT, CD4 or helper T cells and CD8 or cytotoxic T cells. So number one, understand their role. Also very important, understand which T cell goes with which MHC complex. And I'm going to touch on this again and I'll make that very, very clear. Just know for now that that is also something that's important to know. All right, and the last type of cell that I want to talk about in terms of the adaptive immune system are cells called professional antigen presenting cells. And there's going to be some overlap here between cells from the innate immune system and adaptive immune system. These professional antigen presenting cells kind of act as a communication link between the innate immune system and the adaptive immune system. And so the first type of professional antigen presenter is called a dendritic cell, also probably one of the most important professional antigen presenting cells. The second is called a macrophage. And if you remember from the innate immune system podcast, this is a type of phagocyte. It's able to actually engulf and eat pathogens. And now you know it is also capable of antigen presentation. And I'm going to further break down this concept of antigen presentation when I get into the cell-mediated immunity section of this podcast. But in general, it's a pretty simple concept. Essentially what's going on is you have these different cells that are able to actually bind to very specific antigens. And they're just going to bring these antigens to a T cell and say, here you go, I'm passing this on to you. You know, I know it's bad, but I don't really know what to do with this antigen to really stimulate an immune response. So all they're doing is they're passing off this antigen to the cells of the adaptive immune system. It's going to be a T cell. And from there, the T cell is going to activate this big cascade to get everything kind of going. So antigen presentation is just essentially this cell bringing an antigen to another cell. It's a fairly simple concept in general. All right, so that's a few of the cells that make up the adaptive immune system. The two main players are obviously the T cell and the B cell. And so you can break the adaptive immune system into two branches. That's going to be the humoral immune system and the cell-mediated immune system. And so I'm going to start by talking about the humoral immune system, which has a lot to do with B cells and the production of antibodies. All right, so humoral immunity is an aspect of the adaptive immune system that is mediated by macromolecules, specifically proteins that are found in extracellular fluids within the body. And the most important of these proteins are antibodies. And so first I'm going to talk about antibodies, what they are, what they do, and then I'll get into how B cells make these antibodies and kind of all that B cell stuff. I'll get into that next. So in general, antibodies are these Y-shaped proteins that are produced by B cells that can cause the destruction of microorganisms, bacteria, viruses, all that stuff, and prevent the spread of infections. And they do this by very specifically and tightly binding antigens. And so in terms of their structure, as I said, these antibodies are Y-shaped proteins, and they're made up of two subunits. One of these subunits is called the heavy chain, which is the larger subunit of the antibody, and one is called the light chain, which is the smaller subunit of the antibody. And I could explain this all I want and say, okay, on the left side of the Y, that is where the light subunit is. But what I recommend is just going and looking up a picture, seeing where the light chain is and seeing where the heavy chain is, and being able to see that these are two different subunits of an antibody. In addition to being broken down in terms of their structure, antibodies can be broken down in terms of their function. So different parts of the antibody do different things. For instance, there is a region called the Fab region that's essentially on the top of these two Y parts of the antibody that function to bind antibodies. So it's almost kind of like the most important part of the antibody. These are what are very specific and able to bind very tightly to these antigens. In addition to the very fabulous Fab region, there's also what's known as the Fc region, and this encompasses the shaft part of the Y, or like the leg of the Y. And so the Fc region ensures that each antibody generates an appropriate immune response for a given antigen. In other words, this is the part of the antigen that is actually recognized by immune cells. And so a lot of these different immune cells actually have what's known as an Fc receptor that can actually bind to this Fc region. So you can kind of think about the Fab region as being fabulous because it's able to bind to these different antigens really tightly, and it's pretty sweet that it can be able to do that. Whereas the Fc region is the part of the antibody that's actually recognized by our immune cell. So that's kind of the general structure of an antibody. On one end, it has these two receptors that can bind really, really tightly to antigens. And on the other end, it's got this stem that can bind to immune cells. And so in terms of function, antibodies can do a lot of different things. For instance, they can neutralize pathogens, which are called neutralizing antibodies. They can activate the complement system, which we talked about last time, which are these proteins in the innate immune system that are able to punch holes in pathogens in their membrane, in their cell membrane. They also can do this thing called agglutination, which is essentially where they connect foreign bits of pathogens or foreign cells together so that they can be easily phagocytosed into some immune cell. So essentially they're acting like glue to glue all these, pathogens together in a big clump so that it can be easily eaten by one of these phagocytes. In addition, antibodies can use the same process of kind of gluing these insoluble bits of pathogens or antigens together, and they can do this to make insoluble precipitates. So essentially, they can form these big, big clumps that will begin to precipitate and can be easily, or I guess more easily handled by the immune system. And then lastly, these antibodies can activate other immune cells. Still, one of the most important parts of the antibody, and something that I haven't mentioned yet, is what's called the variable region. And this is at the very tip of the Y, of those two prongs. It's at the very tip and at just the tip. And this is the part that actually is able to bind on to antigens very specifically and very tightly. And so what's interesting to me is the process by which these antibodies are produced and able to bind so tightly to a specific antigen. How is it that our body's able to recognize these pathogens that it's never seen before? I could go to the moon, for instance, and let's say that there's this new type of bacteria on the moon. Well, odds are that eventually I'll develop antibodies against this bacteria from the moon, and I'll be able to mount a decent immune response to that bacteria. How does that process happen? Well, this leads me into the next thing I want to talk about, which is B cells. And we'll talk about how exactly the B cells are able to produce this ridiculously wide range of antibodies.
Thanks for checking out the Prospective Doctor MCAT Basics podcast. Sam's doing a killer job taking you through the most important MCAT topics, but what if you need a little extra help? How does a 5, 10, or even 15-point increase in your score sound? Imagine how your chances at admission could increase. Med School Coach's MCAT tutoring can get you there. With the most rigorous selection process of any tutoring company, we see amazing results. We deconstruct each student, find a plan that is going to work, and help execute it. That's why our students add an average 12 points to their score. Completely physician-run and operated, and focusing on nothing but medicine. It's no wonder over 10,000 past students have trusted Med School Coach to get them through the MCAT and into medical school. Check out medschoolcoach.com today and mention code PODCAST for 5% off.
So as I said earlier, B cells develop in the bone marrow, therefore B, bone marrow, bone cell. And during their development, they randomly arrange segments from 3 different genes together to make a unique sequence of genes. So these 3 genes are called the variable gene, diversity gene, and joining gene, which together are called the VDJ genes. And this recombination is pretty interesting. Essentially, you're taking little bits of each of these genes and making this variable region gene. So in the end, you take bits and pieces of each of these 3 genes to make up the gene that encodes for the variable region of the antibody that a B cell produces. And again, it's an important point to know that a B cell only produces one antibody. And I'm not saying that it literally only secretes one antibody its whole life. Like, it doesn't release it and say, "Goodbye, my friend, I'll miss you," and it floats away. Instead, it only releases one kind of antibody. all the antibodies released from one B cell have all the same variable region. Therefore, they only bind to a very specific pathogen. An easier way to think about this is imagine taking a deck of cards and taking just 3 of the suits, let's say hearts, diamonds, and clubs. And now these are going to be the 3 different VDJ genes. And so what you do is you take these 3 suits, and obviously there's a bunch of cards in each suit, and you shuffle them. You shuffle up all these cards, and now you lay them down. Well, each time you shuffle them and lay them down, you're going to get a completely different sequence of cards as you lay them down each time you shuffle them. So in the same way, these B cells, as they develop, create these very unique and random variable region genes through this process of VDJ recombination. And based on some data that I was looking at, this paper claims that because of this recombination process, our B cells are able to produce at least 1 trillion different antibodies based on this random recombination process. 1 trillion, that's a ton. All right, so that's a little bit on how B cells develop and then have this very unique and specific variable region antibodies that they secrete. Let's talk next about how B cells are activated. So first off, it's important to note that the first antibodies that are made by a new B cell are not actually secreted. Instead, they're inserted into the plasma membrane, and they serve as receptors for some antigen while they're stationed inside the plasma membrane. So you can imagine this B cell floating around with what looks like a bunch of peach fuzz all over it. Really, these are antibodies that are kind of sticking out, ready to bind to an antigen may it present itself. And once, in fact, an antigen does bind to one of these antigens that's sticking out of the B cell, let's say, proteins from like an HIV virus or something, then this will actually activate the B cell. And so when you think back to how exactly these antibodies were produced in order to have this like high binding affinity for HIV, as I just said, it's a completely random process, right? It's just this exact assembly of these VDJ genes that produces a protein that's able to bind to this HIV protein very tightly. So it's all super random, but this binding, again, as I said, activates these B cells. And at this point, they begin to proliferate and differentiate. And once they become activated, they become what's known as a plasma cell or an effector B cell. And at this point, they are also capable of secreting antibodies. In other words, they are no longer restrained to just having these antibodies bound to their surface. And so as B cells are activated, they undergo this process called positive selection. So essentially what happens in positive selection is that these B cells that are activated, they become activated because they're able to bind to these antigens, survive and start reproducing faster and faster. And the ones that don't get an antigen bound to them die off. So essentially you are positively selecting for these B cells that are able to bind effectively to whatever antigen is invading your body or whatever pathogen is invading your body. And this works to do 2 things. First, it works to boost the number of cells that are able to bind to whatever pathogen that is invading your body. And number 2, it gets rid of these B cells that don't really serve a purpose, right? If B cells were just able to live forever and, not really serve a purpose, they don't have high binding affinity to anything that really matters, then why keep them around? So I guess you could say it kind of kills 2 stones with 1 bird. And so right now, I probably know what you're thinking. What happens when a B cell is produced that has antibodies that are able to bind to, endogenous human proteins? Say, albumin, like the protein that is able to carry things through the blood, or, hemoglobin. What if it's able to bind to these proteins really tightly? Isn't our immune system then going to think that this is some kind of foreign invader, and then we're going to have an autoimmune response? Well, the answer to that is that B cells that bind self-antigens or, endogenous proteins within the body are negatively selected against. So the body is able to actually kill these B cells that produce antibodies that bind to self-antigens. All right, so let's go back to our example of HIV. Let's say that some HIV peptide has bound to a B cell antibody, activated that B cell. Now that B cell becomes a plasma cell and starts secreting antibodies. These antibodies can then do a bunch of things within the immune system. So they can neutralize pathogens, They can activate the complement system. They can do this thing called agglutination, and I talked about that a little bit earlier, but essentially they're gluing together different bits of antigen or pathogen that can then be phagocytosed all together and destroyed within these different phagocytes. So overall, antibodies are very important for, number one, activating our immune system, and number two, helping us fight infections. And so after this B cell is done secreting antibodies, let's say that we have destroyed whatever pathogen we're infected with, some of these B cells will become memory B cells. And I'll talk about that after I introduce the next part of the adaptive immune system, which is called the cell-mediated immune system. All right, so cell-mediated immunity is based upon T cells, which are created and developed in the thymus, hence the T cells, and they are released into the circulation from the thymus, where they kind of meander around and will enter lymph tissue and then go back to the bloodstream, and then will re-enter lymph tissue back into the bloodstream and so forth. So they like to kind of go into the lymph nodes, back out, hang out, whatever they do. And in contrast to B cells, T cells aren't actually able to recognize antigens on their own, right? They don't have these antibodies that are able to recognize antigens. And so what they need is they need an antigen-presenting cell to bring them the antigen, present it to them, and say, here you go, here's an antigen, check this out. And so the first thing I want to do here is present antigen presentation. I want to describe how this process works. And this breaks down into two different processes depending on where the antigen is. If it's inside the cell, then it needs to go through the Mhc Class 1 process that I talked about a little bit at the beginning of the podcast. If the antigen is located outside of the cell or extracellularly located, then it must go through the Mhc Class 2 pathway. So let's start by talking about the Mhc Class 1 pathway. What happens if a pathogen is able to get inside of a cell? So imagine that somebody is infected again with HIV. Sorry for all the HIV examples here. I just find it a very interesting virus. So say that somebody is infected with HIV. And so what HIV does is it wants to be able to get inside these cells and then integrate its genome into the genome of your cells. And that's because it lacks the appropriate replication machinery to be able to, number one, replicate the virus, number two, be able to produce these proteins that it needs in order to build the full virus, right? You need the GAG protein, the TAP protein, you need the envelope protein. You need all these different proteins for HIV to be able to infect other cells. And so let's just say that in this process, you have some HIV proteins floating around in the cytosol of a cell. And so eventually what happens is a cell is actually going to take these proteins into a proteasome, which is this barrel-like protein complex that contains a bunch of proteolytic enzymes. And essentially it's going to break down this envelope protein or whatever protein from the HIV it has into peptide fragments. And then these peptide fragments are going to be taken to the Golgi apparatus, where they become associated with an Mhc Class 1 complex. Once this peptide MHC1 complex is formed, it's transported to the surface of the cell where it is displayed. And at this point, there is a CD8 T cell or a cytotoxic T cell that floats on by, and eventually it recognizes this Mhc Class 1 molecule, and through a co-receptor actually binds to that MHC1 complex. And essentially, this puts the T cell receptor of the CD8 T cell in very, very close proximity to the antigen that is bound to this MHC1 complex. So it's a little bit like a metal detector. That's how I like to think about it. So if you're my twin brother, this is a true story, you propose to your girlfriend and decide to get married. And what happens is that she's at the beach one day and she has her big wedding ring on that you just bought her, and she takes it off to go get in the ocean, because you're not trying to get in the ocean with a ring, right? And of course, you can guess where the story's going. She loses it. So how do you find that? Well, if you go around and start digging at the beach for a while, right, nothing's gonna happen. You're not gonna find that ring. What they did is they went and bought a metal detector while she stayed at the beach to make sure nobody else was going to come by and do the same thing. And they used a metal detector to search the area where she was at, and they were able to actually find it. So in the same way, a CD8 T cell uses its co-receptor that can easily locate the Mhc Class 1 complex to then put its T cell receptor in very close proximity to an antigen that is presented on the surface of a cell that it would otherwise take way too long for it to find. And that is essentially the basics of how Mhc Class 1 antigen presentation works. And I always remember Class 1, it looks like an I, so I always think I, like it's inside me, I. That's how I remember Class 1, right? Because the antigen is going to be inside the cell before it is presented. And so next, let me talk about how the Mhc Class 2 system works. So say in this example that you are working out in your backyard and you step on an old rusty nail, and that introduces some kind of virus into your bloodstream, into your plasma. And so what's going to happen now is that you have some kind of professional antigen presenting cell that's going to be floating along in the blood, and it's going to come upon this virus and it's going to phagocytose it into the cell. And then once inside the cell, again, this virus is going to be degraded by proteolytic enzymes to generate peptide fragments. And then these fragments are going to be trafficked to the Golgi apparatus, where again, they're going to be combined, but this time with Mhc Class 2 proteins. And then in a similar manner, that's going to make its way to the surface of the cell and be presented on the surface of the cell for a CD4 T cell or a helper T cell to come and recognize. And just in the same way, a CD4 has a co-receptor that's able to actually recognize this Mhc Class 2 molecule, and then that puts the T cell receptor of this CD4 T cell in very, very close proximity to the peptide fragment that's being displayed, which again is an antigen. So you can see that these two processes are very, very similar to each other. The real big difference you need to remember is that CD8 T cells are associated with Mhc Class 1 molecules, whereas CD4 T cells are associated with Mhc Class 2 molecules. I think that's really kind of the big thing for the MCAT. But anyway, so that's a little bit of the basics of antigen presentation. After this antigen is presented to this T cell, it needs a few more things to happen, which I wouldn't worry about knowing for the MCAT, before then it gets activated. So this is kind of the first step in this T cell getting activated, probably the most important step of this T cell getting activated. You know, one of the other steps is that they receive input from cytokines, and these different immune cells also can provide them with signals. And then once activated, like a B cell, these T cells undergo rapid division, and they start to build up essentially an army of T cells. And at that point, they start exhibiting the function of T cells. And they do a shit ton of different things, as I mentioned in the intro when I was talking about the different cells that make up the adaptive immune system. In general, cytotoxic or CD8 T cells will kill whatever cell it is presenting them with an antigen. CD4 T cells, on the other hand, are called helper T cells, and they go do a bunch of things. Most importantly, they can go activate B cells. They can activate other immune cells. They play a lot of different roles within the immune system. I kind of like to think about them as like an offensive line in football, right? Offensive line doesn't get much glory at all, but they play a huge role in the pass, the run, special teams. They have all of these different roles, whether it's pass blocking, run blocking, screen blocking. They have all these roles. They don't get much glory, but without them, a bunch of these other components of the football team wouldn't work out. And so in the same way, T cells are kind of like this do-all cell that really, really helps fight infection in the immune system. All right, so that's the quick gist of antigen presentation and how T cells are activated and a little bit about what they do. The next thing that I want to talk about is the phenomena that occurs once you see a pathogen for a second time. So, say that you get the kissing virus, mono. Say you get it once, well, you're going to get really sick. And, that first time you get sick, it might be for a few weeks. And you got to miss school for a few weeks. And it was probably like in middle school, so it was cool. But the second time you get mono, it's really not quite as bad. And I'm not speaking from experience here, but the second time, It's like 10 times less bad. And so I want to talk about why this is. How is the body able to recognize specific pathogens, long after that first exposure? And this gets at two terms that you should know for the MCAT. The first is the primary immune response, and the second is secondary immune response. So the primary immune response happens the very first time you see a specific antigen. The secondary response occurs each time after that first exposure that you see that specific pathogen. And it's important to note that this first response, or the primary response, is a lot less robust than a secondary response, which is why it takes you longer to deal with an infection the very first time. And so let's go back to our T cells and our B cells. Well, at the very end of an infection, there are some T cells and B cells that have become activated by this specific antigen, and they're still alive and they're still floating around. But this infection is dead. So what happens is that these T cells and B cells develop into memory B and memory T cells. And these memory B and T cells have very long lifespans. They're able to live in the body for, years at a time. And importantly, these cells retain their ability to recognize specific antigens and pathogens. So essentially, these memory B and T cells float around in circulation for years at a time and retain their specific antibodies for B cells. And for T cells, they are able to retain specific responses to these specific pathogens. And then when your body sees that pathogen again, say, same bacteria that you got infected with 10 years ago, these cells are able to quickly amplify and quickly divide and mount a big army. And then because of that, they're able to have this quick, strong response that is known as the secondary response. And so vaccines use this principle in order to provide people with immunity to certain viruses. So there are a few different types of vaccines, but typically they are some subunit of protein from a virus or a specific polysaccharide from a virus, or some of them can even be what's called a live attenuated virus, where essentially they take a virus in its full form and then expose it to a lot of heat or something that's going to kill that virus completely, and then they inoculate you with it. And what this does is it stimulates this primary response, which is then going to produce these memory T and memory B cells. So then the next time that you're exposed to a certain pathogen, you're able to have a really strong and effective immune response and kill that virus before it's able to take control in your body. All right, so I want to finish this podcast by just giving you kind of a big full circle view of the immune system. So let's go back to the very, very first example I gave at the beginning of the Innate Immune System podcast. So you're at dinner and you order the grilled chicken sandwich, and your waiter asks you, how would you like your chicken cooked? And you respond medium rare because, that's how you like it. And so this food comes to the table and, at the outset, you're screwed, right? This chicken has salmonella in it. There's no way you're going to get around this. Of course, you don't know this at the time. And so the first thing you do is you kind of pick up your chicken and make sure that it is actually cooked medium rare. So, you break open your chicken sandwich and start to look at the inside. Well, at this point, your hands have actually already come in contact with salmonella. But as part of the innate immune system, your epidermis has acted as this wall that prevents this salmonella from entering your bloodstream at this point, of course. And so next, you see that, okay, it is in fact cooked medium rare as you requested. So you start to eat it. And as this chicken goes down your throat and into your stomach, it still is blocked from entering your body by the epithelial tissue and the mucus that is in your throat and esophagus. And on the way into your stomach, this salmonella actually clings to food particles. And it does this in order to survive the really acidic conditions of your stomach. And I was really interested in how bacteria like salmonella are actually able to survive in this really low acidity of the stomach, right? Because the stomach is super acidic. You'd think that bacteria aren't able to live in that environment. But what they do is they actually cling close to food and kind of use that food to protect them from those acidic conditions in the stomach. And so anyways, this salmonella makes its way down into the GI tract, at which point it actually can infiltrate through the epithelial cells and get inside the body. That's just a property of the Salmonella bacteria itself. And what this does is at first it sets off the general inflammatory response. And this makes the gut start to get inflamed. It starts to get red. It starts to swell. And as part of this general inflammatory response, cells of the innate immune system that have these pattern recognition receptors are able to recognize the salmonella, specifically portions of its flagella. And then what that does is that actually activates inflammatory pathways, which then leads to inflammatory markers being released, which starts to begin to recruit inflammatory cells to this area where the salmonella is starting to get into your body. At the same time, the complement system, which is part of the innate immune system, begins to be activated, and this starts to punch holes in these salmonella bacteria. And as more and more of these immune cells are recruited to the area of inflammation, some of these cells begin to bring salmonella into the cell. Then they break that salmonella down using enzymes that will break down proteins. And then some of these peptide fragments are then combined with the appropriate Mhc Class 2 molecules, and they bring the MHC peptide complex to the surface of the cell. And then from there, that activates a CD4 or helper T cell, which goes on to start activating B cells, activating other portions of the immune system, and you start to generate this bigger and bigger immune response until eventually you take care of the salmonella and all the bacteria are destroyed and you are no longer sick. At this point, the B cells that have developed these antibodies that are really specific for salmonella, and the T cells that were able to recognize specific antigens from salmonella develop into memory T and memory B cells, so that years down the road, when you're re-exposed to salmonella, you'll be able to have a quick, fast, strong response to this pathogen. All right, so that does it for the Adaptive Immune System podcast. I hope these 2 podcasts on the immune system have helped you kind of see how the immune system works and understand both the innate branch and the adaptive branch. I do think they're important topics for the MCAT. As always, good luck in your studies. Thanks for listening. If you like this podcast, please go give us a rating on iTunes. It helps us in the rankings and helps us reach more people. Thanks.
Thanks for listening to Prospective Doctor's MCAT podcast. If you're a pre-med, you'll want to check out prospectivedoctor.com for tons of free tools, articles, and more podcasts that cover your pre-med life. And if you need help on the MCAT or getting into med school, check out